First, thank you for inviting me to present this survey, which was worked by all 5 operational centers present in South Sudan and Epicentre to look into a concern first from our clinicians and a more global concern. So it's about the prevalence of Plasmodium falciparum HRP2 and 3 gene deletions in South Sudan, and I'm happy to present those results today. I will not talk about K13 deletions, but if there are questions, we can also answer them. So malaria is a big problem in South Sudan, as we all know, with almost 10 million suspect cases in 2022 and 3 million confirmed cases and almost 4,500 deaths. And MSF, it's one of the things we do a lot in South Sudan. So we've been treating more than 300,000 malaria cases in 2022 and 2023. There's a high transmission rate, which is more seasonal in the north and more year-round, although endemic in the south. And as far as we know, it's almost exclusively by Plasmodium falciparum malaria. So why this survey? Well, because the most used malaria diagnostic in South Sudan and in many African countries is the HRP2 RDT, the histidine-rich protein 2 detecting test, which is a protein produced by falciparum only. It's produced in a high quantity, and it allows to produce very solid tests that keep well up to 40 degrees. There's a lot of brands that are WHO pre-qualified. So that's a good reason to have it in a test. Of note that these RDTs also detect HRP3, which is a related protein also produced by falciparum, and this is an important thing to remember. So what's the problem is that there are parasites that start having mutations and deletions in these genes, which were first identified in Peru and have since been identified in many more areas, and especially in the Horn of Africa and more broadly in Eastern Africa. There's not much data of use about South Sudan in terms of the impact of these deletions. And as you can see on the map here, this is a WHO threats map, which has a lot of studies or surveys available in the public domain, but their use is very variable because not all of them have been designed to look at actual impact on diagnostics. But what is known is that in Ethiopia, in certain regions, in Djibouti and Eritrea, and possibly in Sudan, there is issues with this. And the countries I named before have switched to different types of RDTs. So the impact basically is that if the genes are deleted, the proteins are not expressed, and you get a false negative RDT result if the parasite does not express their genes. So why are we not simply switching to other RDTs? It's because there is mostly one alternative antigen that is detected by RDTs. It's the PLDH, the Plasmodium lactate dehydrogenase, which is produced in lower quantities and harder to detect. So there are different reagents that detect multiple species or Vivax or falciparum only. So the biggest problem is they have a higher limit of detection, so they will miss low-level infections. This is why the WHO recommends switching from the current HRP2 test to PLDH only if more than 5% of cases are missed by HRP2 compared to an alternative diagnostic, because you're missing the low level versus missing the deleted parasites. So how was this survey done? It was based on the WHO standard protocol for monitoring of pfhrp2 and 3 deletions. And the main objective, again, was to determine whether more than 5% of affected cases would be missed by HRP2 tests compared to an alternative test. In this case, the alternative test was a p-falciparum PLDH RDT because microscopy was not feasible in the remote sites and with the unstable context. So it's not a perfect gold standard, it's a comparator test that also detects falciparum. And this was done across 10 sites in South Sudan, mostly in the north, northwest, northeast, and 2 sites in the south. So the inclusion criteria were fever or reported fever in the preceding 2 days, and generally children under 5 or under 15 years old were included, and this was purely based on the program in place in the location. So not influenced by the study. Written consent was obtained and assent for the children above 7 years of age. Children with signs of severity were excluded and immediately referred, and previous enrollment in a study was also an exclusion criteria. A brief questionnaire was made, especially on the history of malaria, and both RDTs, the PF, PLDH, and HRP2 were performed in parallel, and a dried blood spot was made from the same capillary blood draw. This was for molecular testing, which was done at the Australian Defence Force Malaria and Infectious Disease Institute. That is the WHO center for this purpose, where they looked at the genes of interest, so HRP2 and 3, PFLDH, and a human tubulin gene as a control. And this was done on all the RDT positives, either or both RDT, as well as a subset of 20% of the negative cases. The sample size, very quickly, was based on a WHO protocol with Bayesian methods and really aimed at being able to tell whether more or less than 5% of cases would be missed, and assuming a 60% HRP2 RDT positivity and that 8 out of 10 sites would be able to participate, which led to the ambition to have 200 suspect cases per site, or 2,000 cases in total. So, on the main findings, 1,842 participants were enrolled between January and March 2024, with a median age of 3. And almost all of them were residents of the county for at least 30 days, except in Malakal, where 37% arrived within the last 30 days. This is relevant in terms of interpreting the data if differences are found, because they could be imported. The RDT results, 40% of HRP2 RDT were positive across the sites with variability between sites, and 32% of the PF-PLDH, so already showing quite a difference in sensitivity. Now for the results, this is a very busy slide, but it's the most important results. So what we were interested to see is cases that would be positive on the alternative test, so the PF-PLDH, and negative on our current test, the HRP2. So 14 of such cases were found, which is 2.4% of the total tests that were positive for PLDH. And when the molecular analysis was done, only 2 of these cases were actually confirmed to be due to pfhrp2, HRP2, and 3 double gene deletion. If you remember, if only one gene is deleted, the other one will still be detected. The others were due to other malaria species, P. ovale, P. malariae, or were negative for PCR, so possibly false positive RDT results. So in summary, to simplify it, less 0.34% of the total PF-PLDH positive tests were negative for HRP2, which is well under the 5%, and the 2 sites where one case was found remained also well under the 5%, even if that's not statistically significant. And of note, none of the cases which were negative for both RDT was found to have a gene deletion, so we didn't miss case that had, for example, very low level of infection. So in terms of the conclusions, luckily the deletions leading to false negative HRP2 tests were rare and well under the recommended cutoff. And for now, the current HRP2 RDT appear appropriate. We can reassure our clinicians, but also this has been informing the National Malaria Control Program and also our own MSF programs. A limitation is that there was a decent geographic cover of South Sudan, but mostly, as you could see, on the northeast and a bit in the south. So other areas might show different results because these are often very localized issues. So follow-up is needed, and a study is currently going on with Armauer Hansen Research Institute in Ethiopia, covering other areas. There were some questions on the RDT chosen for the study, the PF-PLDH, because it detected some other species. So this is something WHO is aware of and following up with the manufacturers. It doesn't have an impact on the study conclusions, which were reassuring. But yes, whether this is the most appropriate test for such purposes. some of the challenges and lessons learned from this survey, which was not as light as WHO was selling it, is that it took about a year of time and efforts to achieve a partnership between 4 parties, so MSF, the National Malaria Program, WHO, and the lab in Australia, which was a huge effort, especially by Epicentre colleagues and Matt in particular, who led this. Of course, supervision in remote and difficult areas was hard, but we have pretty good programs, and this was well achieved. The implementation delay led to missing seasonal peaks, which was not an issue because the results were very clear. The prevalence was very low, but had we been closer to the 5%, we would probably not have achieved a significant result. And on the positive side, the field side of things was relatively easy. To do 2 RDTs and take a DBS with a dedicated person was very feasible. And I'd like to thank our colleagues in Australia, especially Xin Cheng, who did all the testing, Matt, who did a lot of work, or most of the work to pull this together, and Jane Cunningham at WHO, who's been very helpful and continues to be very helpful in this area. Thank you.